This is the topic what I have written, that is describe briefly the synthesis of purine in the body with special stress on salvage pathway. And second point is describe the degradation of purines and its significance. Okay. Today I will discuss about the synthesis of purines. There are three main pathways. One is de novo synthesis, second one is salvage pathway, and third one is another one. Can you hear me? So today I will discuss only the synthesis of purine part, and that out of three parts, the most important part is de novo synthesis part. And then tomorrow, while degradation of the purines, I will discuss about the salvage pathway also. So this is one of the, difficult synthetic pathway. Usually questions are not asked on this topic, but because of the trend, I wonder whether this will come as a long question or not. The names, the enzyme names are all difficult to remember. I have tried to make it as simple as possible, only to make you understand so that you can write something on the answer script, and soon after reaching your hostel in the evening, you try to have a look at the book. Otherwise, you will not be able to remember. Only 2 or 3 slides I will inform you to note down. The rest of the things are available in the book. I have simplified it in a way so that at least you can remember easily and write something. So, first and foremost thing, I do not know how much you have remembered about the purine rings. The DNA nucleotides, you have been told the chemistry of nucleotides, but basically purine rings is a heterocyclic ring. It is having an imidazole group, 5-membered imidazole group, and a pyrimidine group. Okay. So if you remember, this is the pyrimidine group. This is the imidazole group. Now they are numbered in this direction, anti-clockwise. Hey, it is secure actually. This is 1, this is 2, this is 3, this is 4, 5, 6, 7, 8 and 9. At position number there is 1 nitrogen. At position number 3 there is nitrogen. 9 nitrogen is there. Here it is also nitrogen. Carbon is there at C position. It is at C position. C is there. And at 2 position also C is there. So, de novo synthesis means it is starting from the scratch. The entire structure is to be formed from the beginning. It is difficult to synthesize something, and 11 steps are there. The ring synthesis occurs in a peculiar fashion. So this is the right sided ring and the left sided ring. It starts from 9 position. Okay? This ring is produced first, imidazole ring. And after starting this ring, a part of this ring is closed. Then the rest of the pyrimidine ring is formed and then it is closed. So usually it starts from 9 and then you are having 4, then 5, then 7 at a time. Okay? Do you understand? Then 8. After 8th step, this ring starts. That is the 3. And then ring is closed. Next step, this C6 carbon is added, and then it is nitrogen is added, and then carbon is added, and at the last this pyrimidine ring is closed. So this is the sequence, you remember. You start from 9 and then 4, 5, 7 and then 8, stop, come to third position, this is nitrogen, then the ring is closed and then 6, then this is 1 and then this is 2, then finally the ring is closed. Then the total structure is formed. Okay? Now, what I want to emphasize that these structures are produced, where from they will get the molecule? They get molecule from different sources. So start with number 1. See, number 1 is nitrogen. Nitrogen is obtained from acid. পালিয়ে যায় না, যেন রাগ লাগে খুব জোর। দেখো তো একটুখানি কমাতে, একটুখানি সাউন্ডটা একটু কমাচ্ছিল, কমে যাবে। Number 1 is aspartic acid. So aspartic acid is an amino acid, you know. So aspartic acid contributes 1 nitrogen. It is an amino group, it can give 1 nitrogen. And after it donates nitrogen, it is formed, it is converted into fumarate. এই তুমি একটু দেখো তো, ডাকো তো কাউকে, মাইকটা কন্ট্রোল করার জন্য। এতবার ডিস্টারবেন্স হলে মুশকিল আছে, ক্লাসটা ফ্রিজ করতে পারবো না। And the rhythm also get lost. See, so number 1 position comes from aspartate, okay? Then number 2 is carbon. It comes from tetrahydrofolate. You know tetrahydrofolate is famous for 1 carbon metabolism, and 1 carbon metabolism comes as a short note. The 1 carbon is donated by tetrahydrofolate. Folic acid, the active form of folic acid is tetrahydrofolic acid, and it is converted by the enzyme dihydrofolate reductase. So there are several forms, and this form is N10 methyl and then formyl. N10 formyl tetrahydrofolate. Okay? So it gives only 1 carbon. Formyl means this is CHO group. So it gives 1 formyl group here. And this tetrahydrofolate also contribute 1 nitrogen here. This is carbon, sorry. This is carbon. This is N5, N10, methynyl tetrahydrofolate. Both basically gives formyl group, formyl carbon. Okay. So one position is over, two position is over. Now third position contribution is from glutamine. Glutamine is an basic amino acid having an extra amide group. So that extra amide group is given here, and in that process it gets converted into glutamic acid, sorry. And this 9 position is also contributed by glutamine. So third and 9 position contribution is from glutamine, and after donating that extra amino group nitrogen, it gets converted into glutamic acid. So 7th position is there. 7th position carbon is there. 7th position carbon is there. So these 4, 5 and 7, these 3, it is contributed by glycine. C6 is contributed by carbon dioxide. So there are 3 amino acids which are involved. One is aspartic acid, one is glutamine, and third one is glycine. And glycine alone is contributing 3 atoms. And tetrahydrofolate is contributing 2 carbon atoms. Okay. So this is the basic precursors or fundamental molecules, you can say, which will contribute to the formation of purine ring. Now I will tell you in detail. This is a complicated step. So this part you have, I think you have understood this part. Now I will explain everything. Only important part, before I go to show the slides very rapidly, I want to tell you that this cyclical structure is not formed from 9 position itself. Ribose 5-phosphate, which is formed in the HMP pathway, is the starting material. Because purines as such are not required. So purines, there is adenine and guanyl. You know that NAD, NADP, FAD, FADH2, these are used as a coenzymes. Cyclic AMP, cyclic GMP, they are regulators of metabolism. Cyclic AMP, cyclic GMP, they are also, act as a second messenger of the hormone. So basically, we require something in nucleotide form, not exactly as a purine form. So ribose 5-phosphate is readily available in the cytosol. So that ribose 5-phosphate, if it is added, that means for a nucleotide is formed basically. Just if we add to ribose 5-phosphate to one nucleotide, that one adenine or one guanine, then basically one nucleotide will be formed directly. And we will not require one extra amount of phosphate which will be required for ribose 5-phosphate formation. Okay? Is this particular, this is already preformed material which is available in the cytosol, and HMP shunt occurs in the cytosol. And this purine synthesis also occurs in cytosol. So they get the materials readily. But purine, some part are required in the cytosol, rest of the purine molecules are used for DNA and RNA synthesis. So they need to be transported to the nucleus or to the mitochondria, because mitochondria is having DNA, there it is having a protein synthetic machinery, same as just nucleus. So just to enter from cytoplasm to nucleus, it must go through nuclear pore, and there is a complex. There is a nuclear pore complex. You can say, I'll tell you in a very short while that procedure, the structure I will show. Through that structure, it enters into the nucleus. Similarly, through a special transporter, these purines are transported inside the mitochondria. So this synthesis of purine is a very highly regulated process. Why? Because nucleic acids, these enzymes, coenzymes are required for a variety of metabolic reactions, and also for nucleic acid synthesis during active cell division or during growth, or whatever be the reason, there might be variation in the demands of synthesis of nucleotides. So that's why the system has to be regulated very tightly. So this is what I have already shown, that this is the anticlockwise direction, the numbering system. Now why I have shown this thing, adenine and guanine, because this is 6 amino purine is there, this is deoxy. So from adenine to guanine, one reduction step will be required, because here the phosphate will be basically, this is ribose 5-phosphate. So from ribose 5-phosphate, ribonucleotide it form, and it will form the backbone of DNA. For RNA, for DNA we require deoxy. For that we require reduction of the ribose sugar. Now notable purines, we know adenine, guanine, there is no doubt about it. But hypoxanthine and xanthine is also a part of purine, because they will be produced during the synthesis of breakdown, during the breakdown product, as a breakdown product of purine metabolism. And uric acid is the end product of purine metabolism, and that is the derivative of purine. We take carbohydrate, we take protein, we take fat, and these are absorbed from the intestine, and these are used. Unfortunately, nucleic acids or purines, although we take in the diet, these are not directly incorporated into the cell, just like glucose or amino acids. So body degrades it into uric acid, mainly in the liver. Main synthesis occurs de novo, what I have explained. So therefore, since this is synthesized maximally by the de novo pathway, it is dietarily non-essential. And purine, as I told you, its synthesis is a highly regulated process because of the varying physiological demands to synthesize either the coenzymes or the nucleic acids. Purine, since it is a very important biomolecule, nature has evolved several mechanisms to protect it. So purine deficiency state is a very rare condition, and when it occurs, really it becomes troublesome, and it causes only a handful number of diseases, and these are all severe diseases. Like purine deficiency state can occur if there is a folate deficiency, dietary folate, because then dietary folate, tetrahydrofolate will not be formed. So tetrahydrofolate can affect purine synthesis. So these enzymes which I will tell you in detail right now, just note down for the time being, just note down here, this abnormalities of purine metabolism is PRPP synthetase overactivity. Only note down this one. It will result in gout. HGPRT deficiency, I will tell you in detail tomorrow, that causes Lesch-Nyhan syndrome. Adenosine deaminase deficiency, it causes severe combined immunodeficiency state. Purine nucleotide phosphorylase deficiency, it's really a rare disease. It causes autoimmune disorder, benign opportunistic. These are, I wanted to tell you just for relevant, note what the facts basically. Now, nucleotide and nucleoside, you all know the difference between the two. One is having phosphate group attached to it. Now, here ribose 5-phosphate, as I told you, is the starting material. So, we are basically getting at the end is a nucleotide. Now, in this step basically, first step is ribose 5-phosphate is the starting material. Basically here ATP is required and it produces AMP and it contributes to phosphate group. That's why it is called phosphoribosyl pyrophosphate. Ribose 5-phosphate means phosphoribosyl. Pyrophosphate means 2 phosphate molecules are donated by the ATP. Since 2 phosphates are going, that means 2 high energy compounds are gone. So, it is a wastage of 2 high molecular weight compounds. So, this step is catalyzed since PRPP is the name. So, basically it synthesizes PRPP. So, the enzyme name is PRPP synthetase. Just 2 steps you have to remember. Synthetase, why I am telling you? Because it requires ATP. ATP, T is there, so synthetase will be there. All ATP dependent reactions, synthetic reaction require synthetase. And the second step is the PRPP gets converted into phosphoribosylamine. And I told you nitrogen N9 is the first formation. In the first stage, no nucleic acid cyclical ring is formed. It starts from the second step. The phosphoribosylamine is there. These amines comes from N9 position. You just look at the structure. It comes from glutamine. So PRPP will be required. So glutamine will be required. And glutamine will get converted into glutamic acid. And since this is an amide nitrogen, amido nitrogen, so this is called amidotransferase, phosphoribosyl pyrophosphate amidotransferase. Amide group of the nitrogen glutamine is transferred. So first ribose 5-phosphate, then PRPP, basically start with PRPP. Is the starting material, then phosphoribosylamine, So this is synthesized by PRPP synthetase and this is by phosphoribosyl amide phosphoribosyl pyrophosphate is formed by another amido group, amidotransferase group. So these are the two enzymes which you must remember. And then from here, another 9 steps are there totally basically. Finally, IMP, that is Inosine Monophosphate is formed. Okay. So there are a number of steps. This is overview I am telling you. I am not going into the detail. And then from here, AMP is formed and GMP is formed. And from AMP to ADP, ADP to ATP. Similarly, from GMP to GDP to GTP. Okay. Now, see, this is the committed step. Committed step means once this step is crossed, this will finally come to the end product, that is inosine monophosphate. So this is the committed step and this is the first step, PRPP synthetase, and maximum regulation occurs in these 2 steps. That's why I am telling you, you have to remember only these 2 steps. And this is a committed step, means once this step is over, that means it will be produced to IMP. And to produce IMP, 6 high energy compounds are required. So 6 molecules of ATP will be basically wasted. So somehow this structure has to be preserved. Otherwise, again, de novo synthesis will be there, our energy expenditure will be there. So there is a tight regulation at this level first, so that if there is an excess amount of ATP produced, they will regulate the synthesis of IMP. Similarly, GDP is there, that will regulate this thing, PRPP. In addition, through a feedback inhibition, either it will inhibit the PRP synthetase or amidotransferase. Okay? First foremost, from here itself, it will cause inhibition. If excess GMP is produced, it will inhibit this one, IMP. So, indirectly also, it regulates the first step and the second step also. So, they are the regulatory part of this thing. So, basically at the end, we want ATP, we want GTP, which is formed, and when we require AMP will be produced, further reaction will not take place. If you require ADP, if you require GTP, whatever is required, accordingly this will be synthesized. So, this is the overview. Now, we look at this thing here. What is adenine? This I am not going to tell you, because after the ribonucleotide are formed, only one reduction step will be required to make deoxyribonucleotide. That is the last step. So, purine sugar I have noted, and you have already noted what are the different sources of these purine rings. This is important. Now, you have to, what you have to write, I am telling you. You make a chart on your, landscape. You make a chart right now. Only you have to remember this thing, and that will complete everything practically. So, this is not written in your book is so well. I have made it, tailor-made it so that you remember it. Just write a little fast. Then every step you have to understand only, nothing more than the required. And here I will make you understand what are all these things. So write a little faster, okay? So basic material I have discussed. Or otherwise you look down, I'll give you another time, you can, because otherwise if you do not note down, you will not understand. Rest of the things are not required. You just spend at least as fast as possible. Do not take photograph, you note it down, it is there in the book. Book charts are there, only thing is this is not available. And what are the missing points in the books, that I will also note it. That's why I am telling you. You modify this chart after going home, after reading books, you modify this chart so that you remember. I have made it simpler. No photograph should be taken, only things try to understand. It is there, you can write it. Just once you are over, I mean, you have written, just let me know. Many things are to be told. So, during synthesis I told you that 6 molecules, high energy compounds are required. You look at the material here, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. 5 molecules of ATP are there. But 6 molecules of ATP are required basically. Why? Because in the first step, ATP is getting converted into AMP. 2 high energy compounds are wasted. So that is the reason why 6 molecules. Do you get my point? 3 amino acids are required. One is glutamine, one is aspartic acid, and third one is glycine. Tetrahydrofolate contributes 2. There are many advantages of, noting it down. And carbon dioxide, we get it out of 9 steps. You see, there are 2 steps where there are ring closure. This is the ring closure step. At this 6th position, there is 6th step is the ring closure step. Note down now this one, where the ring closes. I told you this purine ring is formed in the reverse direction from imidazole group first, and at the 6th position, 6th reaction, imidazole group ring closes. And at the last stage, 11th step, that pyrimidine ring closes. So now, you remember what are the added atoms you have by this time noted. First compound is, see, in your book, the names will be written very, very long. It will be difficult to remember. If you can remember, it is all right. And the enzymes I am telling you, this 6 synthetase, 7 synthetase, 9 synthetase, these enzymes are not available in the internet. These are written in Harper book. You give a reference of Harper book. In fact, 1988, when I was a first year MD student, I purchased a book. That book is with me. In that book, this enzyme, these stages are not written at all. Even in the current stage, current Harper, this third enzyme is not written. I have taken it from Vasudevan book. Vasudevan book is also, writing something, it is difficult to remember. So since this is written in the Harper book, you can give it as a reference so that you can avoid remembering the names of the enzymes. And the names of the compounds, what I have written here, may not match with what you will read in your own book. Because there will be some alteration, like phosphoribosyl pyrophosphate. Okay? So this is another group. Something I have written, iminoimidazole, something like that. So that name will not match. Here you write down, these are the red points you note down, just write down and underline it so that you remember. So first step is phosphoribosyl pyrophosphate. Pyrophosphate you underline, and then second step only amine group is attached, this amino is amine, just underline only amine. Third stage, glycine is attached, that's why it is glycinamide. So glycinamide is added only, this part, ribosyl 5-phosphate is there. One point I want to highlight, most of you are having, Dr. D.M. Basudevan's book I have seen, they have written ribonucleotide. Ribonucleotide means a complete purine base is to be formed, which has not formed at all at this stage. But in the stage, you will find the name of ribonucleotide. I do not know why it has been given, but this is not, ribonucleotide was not formed at all, though no purine was formed at that stage. Yes, purine formation will be complete after inosine monophosphate only. So still the name is there. So instead of ribosyl 5-phosphate, you will find the name of ribonucleotide. Now here you see glycinamide, then methynyl tetrahydrofolate N5N10, it contributes one formyl group. That's why only you add this glycinamide, ribosyl 5-phosphate is there, only formyl you just prefix it. So that stage is over. And then formyl group is added, and then extra amide group is added, amide group. That's why it is called formyl glycinamide midine, not mide. First stage is mide, second stage is another amino acid. So that's why it is called glycinamidine. Remember, don't get confused. Look at the book very carefully. You will see the difference in the spelling. Now, then you get a carbon dioxide. So there will be a carboxylase reaction. So you get a carboxylase. So amino imidazole is there. So ring imidazole group is closed. That's why its name is changed to amino imidazole. Okay. Now here carbon dioxide is added. So only carboxylate term is added to it. On the amino imidazole ribosyl 5-phosphate, here only carboxylate group is attached. Now here there is another amino acid is added. That's why this aspartic acid is still attached to it. That's why its name is succinyl. The same structure, succinyl. Now when this aspartic acid is deaminated, means when it gives the nitrogen, it gets converted into fumarate. So fumarate is removed, and now you see the name which has been changed from succinyl, it becomes carbamide. It is no longer carbamide, this amino imidazole carbamide 5-phosphate succinyl part is over. Now again one formyl group is attached here. So the name one formyl you have to attach here, formamino. And then ring closure occurs, that is IMP monophosphate. The advantage of this is that you need not remember all 11 enzymes. And in Harper book, only 10 enzymes are written. This third enzyme is not written. And those in Dr. S.K. Gupta, I have checked the books. This step is not written. He has simply copied from Harper. So you have to remember these first two steps and this step also. Glycinamide, you see, remember, glycinamide 5-ribosyl 5-phosphate is formed here. So basically this is synthesis. So you will get glycinamide ribonucleotide synthetase. What I am telling you, see the name here, ribonucleotide. I do not know why the name has been given nucleotide, because nucleotide is not there, not yet formed. The formyl transferase, there are two-step formyl transferase. For easy remembering, you can remember formyl transferase. Now, if you this is not difficult, What is glycinamide ribosyl 5-phosphate formyl transferase? Because glycinamide is a precursor molecule to which we are adding formyl. So, glycinamide ribosyl 5-phosphate formyl transferase will be the name of the actual name. Similarly, here amino imidazole carboxamide ribosyl 5-phosphate formyl transferase. Okay, it's like that. You see the only for your convenience, at least you remember that 6 synthesis, 7 synthesis, 9 synthesis. So, synthetase is here 1, synthetase is here 2, synthetase here 3, 4, 5. 1, 2, 3, 4, 5. And last stage there is a requirement also. You see there is one more, this structure. This structure synthetase is there. This structure ATP synthesis is there, synthetase is there. A ring closure is there, one part. Coming to this part, what I have discussed. So, briefly there are 3 main parts. One is de novo synthesis, which is the main pathway, and there is ribosylation of purine. Purine means it is only purine molecule is there. So, if you want to make nucleotide, we have to phosphoribosylate. Means you have to add phosphate, you have to add ribosyl. So, that is phosphoribosylation. But if you add a purine nucleoside, we use a precursor molecule, then we have to add only phosphorylation. Okay. So, as I told you, there are 11 steps, and the 11 steps names of the enzymes I have shown it to you. Also the 11 enzymes, and finally inosine monophosphate is the form. And from inosine monophosphate again AMP, GMP to GTP. Now at one stage deoxy adenosine or deoxy DATP and this GTP will be required, because this will be required for the DNA synthesis. So this is HGP, this is salvage pathway, I will discuss tomorrow. So now open the chart. Try to remember, this is what I have told in the beginning. What groups are required? See, remember how the way I have told you, so that you can remember. Else you can remember starting material is from 9. Remember, imidazole ring is formed first from this anti-clockwise direction. Then C4, C5, 9, 3 molecules are contributed by glycine. Then C8. You see that is still imidazole group. N3 is in pyrimidine group, pyrimidine ring. So N3 is added, then ring closure is there. As I told you, after almost completing the imidazole group, first step of pyrimidine ring is done, and then ring is closed. Then you see the added on the top, that is carbon dioxide is added, and then aspartic acid is added, and aspartic acid contributes 1 nitrogen, ammonia, and then because of deamination it gets formed into fumarate. And then at the end C2 position formyl group is added. When you write down 1 carbon metabolism, you write at least C8, C2 of purine rings are contributed by tetrahydrofolate reactor. They are tetrahydrofolate, hydrofolate, okay? Tetrahydrofolate, contribution of one carbon pool. And then finally ring close is done. Now this is ultimately, you see the carboxyl, here you note down one thing, if you have noted it down, you in the 7th step it is carboxylate, and in the 9th step it is carbamide. You just underline it so that you remember the difference between the two. So this is what I have told you. 3 amino acids, glutamine, glycine is required. Here you see, this is this stage. And carbon dioxide is added here. Methynyl is group is required at 2 steps, where methynyl or tetrahydrofolate is required, formyl transferase enzyme is required. So what I have said that you have come now I am telling you, individual components as fast as possible. If you cannot remember all these things, there is a shortcut for you also. So you write down first step, ribose 5-phosphate, first 2 steps, these are the most important steps where maximum regulation occurs. Then you add 5%, just whatever you remember, you just write down everything and then you bring it to inosine monophosphate. That is the simplest way. If you cannot remember by chance, because there you have to remember many cycles, many things, at least write down this first 2 steps and whatever you remember, you add to it. Don't forget to add ATP. And then you make IMP, from IMP to AMP, GMP. Okay? Only remember that the first step is catalyzed by the PRPP synthetase, and this step is the commutative step I have been repeatedly telling, that once this stage is over, it will get converted into IMP. And body tries maximally not to destroy IMP, that I will tell you tomorrow. So this same pattern, now individual pathway I am telling you what I have told you, ribose 5-phosphate. So PRPP is it is formed, so it is called PRPP synthetase. Here you note down, ATP is getting converted into AMP, means 2-pyrophosphate. So this is not contributing anything to purine synthesis, but 2 high energy phosphate bonds are required to add nitrogen atom at the first position. That is the main purpose. In the second step, this pyrophosphate is removed. Because the energy is required to add nitrogen atom in the first position, the phosphoribosyl pyrophosphate is added. Otherwise, there is no requirement. See, wrote down here hydroxyl group is there, 2 phosphates are attached. So ribosyl PRPP synthetase is the enzyme. Now you see that part is over. This pyrophosphate was there, pyrophosphate is now out. Instead, this nitrogen has come. This nitrogen is the N nitrogen, imidazole ring. Now you try to understand this thing. This you will get in the book. Only the arrow you may not get it. I am showing it to you. Now this first step occurred. Then I said that this will move in this direction. So glutamine PRPP amidotransferase is the enzyme, and in the second stage, glycine is added. So glycine, you see, look at this thing. NH was there already pre-existing, onto which CO, CH2, NH. 3 atoms are added in this structure, this part. So it is moving in this direction. Now N5, N10 tetrahydrofolate, because carbon group is to be added at the C8 position. So this C8 is coming. This, can you see this one? Now, although it is shown at this stage, ring is here, it is closed, but you see ring is not closed here. Ring is not closed. Do you get my point? So this is the enzyme. Now you see the imidazole group. I told you after partially almost completing the imidazole group, first step of the pyrimidine ring structure occurs, and that is the NH. NH is this basically 3rd position nitrogen. Then you see the ring closure. This is the ring closure step. Do you get my point? So that part, one part, the imidazole ring is formed. And now this N nitrogen is still attached to it. So 6th step basically is 6th and 11th stage are for ring closure. Now you see, once this stage is over, I told you that C6 will come. This is carbon dioxide. This part is over. Now from here it will come in this direction. So carboxylase enzyme is there. So you can write down that 5-imidazole ribosyl 5-phosphate carboxylase. So that will basically, that will be the name of the enzyme. After this stage was over earlier, carboxylation, then first nitrogen is coming from aspartate. It is still attached to it. That is the reason why its name is succinyl. In the next step, you see the next step, the succinyl group, that aspartic acid group is out. Only nitrogen is there. Amino group is there. That's why fumate is removed in this state. This name is, the enzyme is adenyl succinate lyase. Means there is no water requirement. So, another name is adenyl succinase. Simple. These are synonymous. When you are in doubt about any enzyme, you check the EC number. Whatever was the name of the enzyme, the EC number will be the same. So, there will be slight alteration in whatever I have told you. The name of the enzyme might be different because one name enzyme is named in by other names also. So, last stage is formyl tetrahydrofolate C2, and now it is almost completing the, this is a formyl group, that one carbon is always formyl group. Only thing that depends what type of tetrahydrofolate is contributing, whether it is N5, N10 or it is N10 or it is N5, whatever it is. If you cannot remember N5, N10, write down tetrahydrofolate. At least that will serve the purpose. So, these are the metabolic inhibitors I have told you. I will tell you in tomorrow also. So, their 10th step is they are almost complete, but the exact structure of purine is not formed. So, it is formed by the last stage, that is one water molecule is removed, and that is why this bond is now purine formed. Total purine ring is finally formed. So, this is called cyclohydrolase. In some books you will get IMP synthase. IMP is there. So, IMP synthase and cyclohydrolase are the same things, same enzyme. If you check the EC number, you will get the EC number, same EC number. Only the name is changed. That's what I told. In many books, you might get a slightly different name. If you are having a little bit doubt, check Harper, it is a reference book, and you check the EC number. So, this is a 6-membered pyrimidine ring closure. Now, this is complete synthesis. Okay? AMP to IMP. Only one step is there. Inosine monophosphate to AMP is formed via one step, that is চলে এসছে স্যার চলে এসছে ছেড়ে দিচ্ছি ঠিক আছে কালকে বলবো এইটুকু বোঝা গেছে তো আজকে গিয়ে বইটা চট করে খুলে নেবে তাহলে মুখস্থ you will not be able to really memorize. It is difficult. Today you make your chart in your own way. You just go on adding whatever chart I have made from your book, whatever you have read, and make slightly short note.